discussion as well. Okay, so we're starting recording. So welcome, this is a review session for our first exam, and I'm basically here to answer your questions about the exam. And the first question that I had was, will every single question that's on the study guide be on the exam? And the answer is no. That's a lot of information that I've asked you to study. So not every single question will be addressed by some topic on the exam. But as I mentioned in lab last week, we should focus on the three main topics that I outlined as possible open response questions. Those topics are also covered in the short answer topics below in your study guide. Okay, so just put that in writing for those of you who can't hear me. So other questions? Do you have questions about specific points on the study guide? And I will actually put that up. Just going to enable you to see my screen after I open up the study guide. In the meantime, you can go ahead and either state if you have audio or type in any questions you have. Kiara, do you have any questions? Hello?
Hello?
Hi, who's here?
Kiara?
Kiara, great. I can hear you. Wonderful.
Okay, so I can hear you and see you now. All right, cool. Okay.
Awesome. So it takes a few minutes for me to kind of set up and make sure everything's working, and I'm glad to hear your voice. It makes it easier to respond to your questions. I'm just opening up the study guide because Kelly had a question about it, and I'm going to share my screen.
Okay.
Kelly Weston will stop sharing content as soon as you start sharing. Okay. Oh, Kelly, were you trying to share something? Okay. Let's get rid of this. All right. I'm going to open up. Oops, that was it. Okay. So Kelly had a question about every topic that I've listed on the the study guide here, and as I mentioned, I'd say know these first 3 topics under open response really well. And there's some related questions here in the possible short answer. I can't possibly ask you all of the questions that I've listed here, just there's not enough time on the exam. But anything on here is fair game. And certainly I've emphasized by the questions I've selected, either on the reading quizzes or the visual quiz, or the animation quizzes, what topics I want you to know in more depth. So that should be also a guide, the homework that you did for preparation for the exam. There were some questions that were not on the homework that are in this bullet list that I'm giving you of the study guide. So Kiara, did you have any specific questions?
Just like, I'm going through the study guide now. I was trying to get some before the webinar started.
Mm-hmm.
I didn't quite get down to trying to find the answers on my own yet about the 5 basic stages of infectious disease.
Okay.
I don't remember stages. Usually I like jot down the things, so I didn't do my own research yet. But that one and the 5 key factors that contribute to the spread of infections, are those like the key factors like that, like the different transmissions, like the active transport and passive transport, vectors and that sort of thing? Are those big 4
right.
key factors, or is that is there something more specific I should be looking for?
Okay, so a good place to look for that is in the slides that I have for this chapter. So that would be from Chapter 2 material.
Chapter 2.
Actually,
yep.
Let me close this. So in the Chapter 2 slides, I've given you a couple of examples, of the stages of infectious disease. And let's see, where is that? So here, the stages can be divided into these 5 categories of, like, first there's an incubation period where you don't actually know you're sick. There are no signs or symptoms. Then there's a prodromal phase when you have, like, kind of maybe generic symptoms that may be common for a lot of different illnesses and diseases. But then you really get sick and you really have, like, clear symptoms and signs of disease. And then it kind of peaks here. And then there's your immune response. So this is time on the x-axis and number of microbes on the y-axis. And so with the peak of number of microbes is really when you feel the worst. And that's when your immune response hasn't kicked in yet. You have, of course, immediately an innate immune response, but your specific immune response that's responsible for clearing the pathogen has not occurred yet. And so then it's starting to gear up during this period of illness, but at the peak is when it starts really winning the battle, where the pathogen is no longer able to grow in your body as a host. And there's a period of decline because your immune system is clearing the microbes that are causing the infection. And then there can be, so that either there's convalescence in which you don't have any symptoms and you recover, or in the case of a chronic infection, you may have periods where these stages of disease repeat. Okay, so those are
an example of that one would be like HIV?
Yeah, HIV is more of a slow, so you have an initial a period of illness, and then there's a period of convalescence, and then yes, you could then see, you would see part of the 5 stages. In the case of AIDS, which is, because it's terminal, it would never, well, each disease would have, that you got as a result of AIDS, would have these 5 stages of disease until one of them caused the death of the patient, right? So let's say you got thrush and then you recovered, you would have the 5 stages of disease again for whatever, it would not be HIV directly, because AIDS is a collection of diseases that result as a because your immune status is impaired by the presence of the HIV in your body, affecting your immune cells, your T cells. But something like herpes, where you have, these, for oral herpes, you have these kind of outbreaks where you get sick and you have these lesions on your mouth, and then it goes away and you have no signs. And then it comes back, you have an outbreak again, or an episode. So that's kind of an example of a chronic disease where you would have these five stages.
Where would you classify like chickenpox and shingles on that? So I know that I heard that chickenpox, it like lies dormant and then eventually comes back as shingles. Is that
not all the time, but it can.
Yeah, not all the time.
Yes.
Would that be the last, I can't pronounce it, the convalescence?
Convalescence? No, convalescence is when, so for example, you get
it's more like the
chickenpox.
herpes virus? Okay, I'm sorry.
So convalescence occurs whether you have a chronic infection like herpes, or whether it's a slow, persistent infection like HIV, or whether it's the varicella virus that affects the chickenpox, causes chickenpox. All of those would have these stages of disease, including convalescence. But there are different set of epidemiologic terms that are used to describe those diseases as being acute. Like in flu, we get sick, we get over it, that's it. You're done. It's not going to come back unless it's a different strain of the flu or a cold. Once you get it, you're immune for life. But there are 100 different cold viruses that you can catch, so usually you get a cold more than once in your life, right? So those are different terms, but these stages of disease are really stages that we see with all diseases. So the convalescence is when there's, as you can see, the number of microbes on the timeline dips down below what we can detect, or it's completely cleared from your body and it's not there anymore. Is that clear?
Yes, it is.
Okay. So your book has a nice bar graph that shows both the number of the microbes and the immune response. And you can see the yellow or gold bars are the number of microbes. You can see it peaks first during the illness phase, but then once your immune response is up and running, and this is really the specific immune response, like things like antibodies and T cells that take time to be generated in response to a specific pathogen. And so you see the blue bars take a little bit longer to peak than the gold bars. And when the blue bars peak, the gold bars are reducing because your immune response is really effectively clearing the pathogen from your body. Yeah.
That makes sense. I think one of the more weighted questions would be a little about the, well, before I get there, I think I could, if I could find it on my own, the 4 key factors of contributing infection, that's the vector, that's the one that it's active transport and passive transport. It's one of the 4 key factors that contribute to the spread of infection. When I think of spread of infection, I think of like the hand washing. I'm thinking of passive transmission and active transmission.
Yep.
But those are 2. What are the other modes of transmission?
Okay. So
or is it specific within each one? Because it says there's 4 key factors.
Let me see what I was talking about. Okay, so the spread of infectious disease. So this actually, I don't think I have a slide for that, but in your book, it's you have to have a susceptible host, right? If we're all immune, right, we all got vaccinated for polio. So if there's nobody that's susceptible to the pathogen, then there will not be any disease, right?
Right.
So that's one factor.
That's part of the postulates, right?
It
one
does
he has to have a susceptible host to infect something with. The third part, or no?
Yeah, that's related to it, yes, in order to test whether a specific microbe causes a disease, then it certainly you have to have a susceptible host to test it in, right?
Okay.
So that's related to the factors. So susceptible host is one thing. You also have to have the right route of transmission. So if it's a sexually transmitted disease, but you're drinking the virus, then that's why we can't catch HIV by drinking from the same cup as somebody who has HIV positive, right? Because it's not the right
portal of transmission.
Yep. Right.
Okay.
And then, so it does have to do with transmission also. As far as susceptible host, you have to have a reservoir that you're exposed to. You have to be exposed in some way to the pathogen. If it's, like Ebola kind of hiding out in the jungles of Africa and you don't go there, you're not going to catch Ebola, right? Those kinds of things. So you have to think about the reservoir.
So the reservoir is where it's inhabited?
Yep, but it can be another animal or human. In the case of poliovirus, it only infects humans, so we don't have to worry about getting sick from contaminated water or from a mosquito bite. It's just humans, right?
Okay.
So a reservoir could be an environmental reservoir like food or water, or it could be also an animal. You get a bite from an animal, you could get rabies virus, or a vector like a mosquito or a tick or some kind of insect. Yeah.
Okay. And what was the 4th key component? So we talked about susceptible host, the right portal, and the reservoir. What's the 4th factor that would contribute to the spread?
4th.
What's the agent? I know the reservoir, we just explained that, and then the host that's to me is obvious. But the agent, is that?
That's the pathogen itself.
Okay. It's the microbe. Okay.
So yeah, the agent is always the microbe that causes the disease. Sometimes we know it, sometimes we don't. It's a new and emerging disease and we're not really sure what it is. But in epidemiology, that's the point, is to use postulates to try to identify the agent that causes the disease, the signs and symptoms of the disease. And that's a microbe. It's always a microbe in the case of infectious diseases. And for the most part, we're talking about human hosts, but sometimes the disease, the pathogen itself or the agent itself will have more than one host. So you can think of like Zika virus. Okay, the host is humans, but it's probably also some animals. It's carried by mosquitoes, but mosquitoes it doesn't make the mosquitoes diseased, so that's not a host. A host is where it shows some kind of symptom as far as, it kills the animal or it makes the animal sick.
So is the mosquito in that case the reservoir?
It is a reservoir, as well as the animals that it infects them.
But it's not a host. The host would be the human or the animal that the mosquito infects.
Yeah, right.
Okay.
It's a vector, so it's more like there's a triangle of that epidemiologists, let's see, here we have it, the triangle of epidemiology that so this triangle of epidemiology in the study of epidemiology, the goal is to identify for every disease an agent. The host is always a human, but it can be other animals as well, and the environment or reservoir. And then we also want to understand how it's transmitted, how it's passed, the agent is passed from its reservoir in the environment to a host, or from host to host, if it's a communicable disease. Not every disease is communicable from human to human.
Starting to piece together most of what wasn't making sense. I think what makes me most nervous about the test would have to be the gram positive and gram negative. You did a pretty good job, though, explaining during class about the staining process and the layers.
Yeah, I thought that was a good animation because it showed you kind of what was happening. I mean, basically the visual quiz that I gave you also showed where the stain would be in each step, but you should be able to fill out that that table in your lab manual. That we have it available electronically.
Is it the bacterial cell envelope? The one that's labeled bacterial cell envelope?
No, it's a table that just asks you at each step what color the bacteria are. So let's go to the lab manual, and this is
on
available.
Our lab manual. Okay.
Yeah, it's in the lab manual itself. So you have to go to the electronic copy on Moodle.
Okay.
I think I have. Well, I can go.
Do you have a link to the video that we watched on during lab?
Unfortunately,
the video helped. No.
Yeah, I know, it's really helpful. Unfortunately, that's something I have access to because I have an account with another publisher's website. We used to use that book, so access used to come with the textbook that we used to use, but we don't use that one anymore. And this textbook, it has some good videos, animations, but not a lot. So unfortunately, that resource isn't available. I'm just gonna see on our website, if we go down to the current week, just last week we have gram stain. Here it is. So this is the So you should understand, like, the lab actually gives kind of nice background in gram-negative, gram-positive, and why it's important medically, because right away the physician, without even knowing what the pathogen is, just knowing is it gram-positive or is it gram-negative, will be able to start treatment, which is, you know, critical as far as time. You know, it would take a lot longer to identify exactly the species that's causing the infection. But because this gram staining procedure, which takes, you know, in an automated machine, which is how they do it in the diagnostic lab in the hospital, it takes a matter of less than an hour to get a result. You guys are doing it by hand, but they don't do it by hand anymore in the hospital. They just have a machine that they make a smear and they stick it in the machine and it does all the staining steps automatically. But this is the steps that I want you to understand as far as, this didn't copy really well, but it's a step. So first step is crystal violet, then Gram's iodine, then ethyl alcohol, then safranin. So what color are the cells? So if it's gram positive or gram negative,
they're both going to be violet.
They're both going to be purple, yeah. Yep. Because you're adding the purple color. It's just like a simple staining procedure like we did last week. It colors any cell, right? Then the Gram's iodine doesn't have color in itself. It's what we call the mordant, okay? So still purple, purple. Mordant is like basically making the dye physically bigger, so it's kind of trapped inside the cell. And then the ethyl alcohol is where you see this differential part of the differential staining method, right? So gram staining is a more complex method that shows a difference between two groups of cells, whereas simple staining, whether it's gram positive or gram negative, is always going to just turn the color of the stain you use. You won't be able to see any difference in the color at the end of simple staining. But so that's where one of the cells, so which one is being decolorized, the gram positive or gram negative?
The gram negative will be clear.
Yeah, right. So you'll have purple for gram positive and clear for gram negative. And then the safranin is just to give the gram negative a color again so you can see a difference. So you still have purple. It does stain the gram positive like we discussed in lab.
Yeah, but it doesn't.
But it's not dark enough to change the purple color, so it's really just purple is positive, red is negative in the end. And that's a really important distinction because a lot of this is reflecting a difference in the structure of the two groups of cells, right? But that structure is the cell wall structure. And we know that lots of antibiotics target the cell wall and are able to kill bacteria that have a thick layer of peptidoglycan, like gram-positive bacteria, but not the gram-negative bacteria, because they're protected by the outer membrane.
What exactly do we need to focus on when studying the cell wall composition of the differences in the grams? I know that the peptidoglycan is a huge difference, and that it's like LPS, and that the fat, the lipid, turned into the endotoxin. What specifically do you want us to know to concentrate on for that?
Okay, so let's look at Chapter 5 lectures. Chapter 5. So Chapter 5 is really what goes, Chapter 3, you're introduced to the difference as far as the staining procedure between gram-positive and gram-negative, and they just gloss over, okay, this has to do with the cell wall being thick or thin, right? But in Chapter 5 is where you really learn the detail of the cell wall. So let's look at that. Okay. So the cell wall is this outer structure beyond the
what page is this on the slide?
page 20, I think it's, what is that?
Go down just a little more
Oh,
on
yeah.
the bottom.
6.
6. Okay, thanks.
Yeah, so it's talking about peptidoglycan. So the
Thank
cell
you.
wall in bacteria, but not in other microbes, not in fungi, even though they have a cell wall, it's not peptidoglycan. Archaea, even though they have a cell wall, it's not peptidoglycan. And algae also have a cell wall. It's more like cellulose, not peptidoglycan. So this composition, this chemical, this polymer of sugars, of the N-acetylglucosamine and the N-acetylmuramic acid, are unique to bacteria. The cell wall has an important function, whether it's composed of peptidoglycan or cellulose. It's going to have a function of protecting the cell from lysis, right, like we talked about with the osmotic pressure. But in the case of peptidoglycan, I do expect you to know the glycans that it's made up of and the key differences. So NAG, NAM, the N-acetylglucosamine and N-acetylmuramic acid. That is alternating chains of those long chains covalently linked. That there's a lot of these layers of peptidoglycan in the gram-positive, but not in the gram-negative. Maybe one, maybe a few. And you should know what's in the gram-positive versus gram-negative. We can have that later. Okay, so gram-positive versus gram-negative. This is on page 7 of the slides. Okay, so the organization is right outside of the cell membrane here. You see these many, many layers of peptidoglycan. There can also be an S layer and also other things, but focus on the cell wall. This is a matrix, like I mentioned. It's kind of like a sieve or a filter. It has a lot of space in these many, many layers, so it's open for any molecules to enter, including antibiotic drugs. But basically, there's a machinery, a huge complex of proteins that are constantly working on the cell membrane to make more of these glycans, these long chains of these two sugars, NAG, NAM. So that is very different from the anatomy of the gram-negative cell wall, which has just one of these or two of these layers of peptidoglycan. It has a large space that's called the periplasmic space, and then it's tethered to another membrane. It has two membranes around it. So remember that the cell wall is a wall. It's not a membrane. So when you hear membrane, that should trigger certain biochemical properties, like membranes being selective and containing lipids, not sugars, right? So the wall is made of sugars, and that's true of, cellulose is a carbohydrate, right? So it's true of fungi, which have chitin, which is another type of carbohydrate or a glycan. But in the case of the outer membrane, that's a lipid bilayer, two layers of lipids. It's different than the cell membrane in that it has LPS, as you mentioned. And this is key for the damaging effects that can occur as a result of a gram-negative infection. So LPS is composed of just lipopolysaccharide. It's part lipid that is intermixed with these traditional phospholipids that you find in the cell membrane, and it's part polysaccharide or carbohydrate, so a long chain of sugars. The sugars are often slightly different in different strains of the same species. So like E. coli can have, there are different pathogenic forms of E. coli that can be distinguished based on their O polysaccharide, or this part of the lipopolysaccharide that's that has a different organization of the sugars. So you might hear, oh, there's been an outbreak at Chipotle, it's E. coli 0157. That's the part of the SBS, that's the sugar part that they're talking about, that we can actually trace and figure out if somebody's sick with food poisoning, whether they were sick because of the same strain or different strains. But you can always kind of take a case history of the patient, where did you eat, and kind of get an idea. But when you actually test the pathogen that's making them sick, so you take a stool sample and you test it for which O antigen is present, you can actually prove and link it molecularly of that strain to, and then you have to have a sample of the food. And if the food also has that same O polysaccharide, then you know where the source of the outbreak of food poisoning came from. So it's useful in following the source or determining the source of an outbreak. But other than that, it's not damaging to us as far as the infection happens. That's the lipid A, or the endotoxin portion of it. That's damaging, and it causes a rapid inflammatory response, which can lead to a big drop in your blood pressure and other inflammatory responses that cause damage to your healthy tissue. So in the case of, there are a few key differences that I'd like you to know about besides just the thick versus thin. Because there aren't many layers in the gram-negative, you're not going to find the it's not marked here. You're not going to find the peptides linking the peptide chains linking the gram-negative cell wall, but you will. Oh, here it is. These tetrapeptide chains will link the many layers of glycans in gram-positive, but it's just forming a link with the outer membrane in the gram-negative. So it's not as strong, but it is more selective in the gram-negative. And the teichoic acids are also not are present. They kind of these long polymers that make up the teichoic acids are the blue lines here that go all the way through the wall from the cell membrane, are present in gram-positive, and they give the gram-positive a kind of net negative charge, which actually helps when we go to do the gram staining, because it's a positively charged dye, and so it actually helps attract the dye and makes it easier to stain. So I hope that answers, I want you to know LPS, lipid A, O polysaccharides, and the cross bridges.
And also,
to
study more.
Yeah, also these key differences in response to antibiotics. So penicillins or vancomycin will be targeting the gram-positive cell wall much more effectively than the gram-negative. Lysozyme is similar, but it's not produced by other microbes. It's actually produced by our own body as part of our innate immune response. We have in our secretions, like in our saliva, in our tears, we have lysozyme that helps protect against infection because it breaks the already intact cell wall. So the antibiotics only prevent the production of new cell wall. They don't break down the existing cell wall. So The cells have to be dividing and growing in order for it to be really effective at killing the cell, the bacteria. But the lysozyme will work whether the bacteria are growing or not. And your book mentions about the resistance that can occur because either the bacteria can just, they have a pump that can be used to quickly remove the drug from the cell, or they have these enzymes that they make that cleave the antibiotic drug, the ring structure, so it's no longer able to bind its target and prevent cell wall synthesis. So the target enzyme is transpeptidase, which is part of this large complex that's necessary for cell wall synthesis.
There's another question that says, how does motility and chemotaxis enable bacteria to reach? What is chemotaxis?
I forget, where do I have it? So chemotaxis
motility is movement, correct?
Motility is movement. So there are basically two ways that bacteria can move, unlike
flagellum, I know is one of them.
Yep, flagella or flagellum. Yep.
I know pili and stalk, those cause like stickiness, right? Adhesion.
Yeah, yeah. So pili, they can, if the entire bacterium is covered in pili, it can aid in attachment, but it can also be a kind of this synchronized beating of the pili that lead to gliding motility. So it's fast as the motility that a flagellum would afford a bacterium, but it can help the bacteria move, and sometimes they move even by just growing together in a community and secreting enzymes that break down the substrate that they're growing on, and literally kind of sliding through the slime that they create as a result of breaking down, secreting these enzymes that break down the things in their environment. But really the chemotaxis is referring to the flagella or flagellum. And that, I thought I had a couple slides for that. Let's see. Do I? Grouping. Oh, here it is. Yeah. So it's on page, there's an image, an electron. On page 11 it starts. So bacteria can have one or many flagella. If they have one, it's a flagellum. And they have this molecular motor that drives them in to beat in a counterclockwise direction that moves the cell. But they have to kind of reset, so they often will pause and not move, and then the flagellum will rotate in a clockwise rotation in order to reset, and then it will continue to move. So what happens during the time that it's resetting is it kind of tumbles or flips over and doesn't have really any specific direction that it goes to, but chemotaxis is a movement up or down a gradient. And the gradient can be a chemical or a nutrient, like food, where it would want to go towards that high concentration of food, or it can be a toxin, like an antibiotic, in which case it would want to move away down the gradient to the weakest point, the lowest concentration of that substance, right? But whether it's moving toward, up a gradient or down a gradient, like toward an attractant or away from a repellent, the movement is the same. It's always counterclockwise movement helps move the cell, and then it tumbles during the clockwise rotation.
There was one thing that I've been trying to get a couple of things. So with the textbook and the notes refer to that bacteria, the size of bacteria, 0.2 to 2. And
yeah, I'll say about 1 micron. No, it's micrometers.
Micrometers.
Yeah.
That's what the funny-looking U stands for? Was micro?
Yes. Yeah.
Okay, not to be confused with the MM for millimeters, I think. Okay, I get that. Okay.
Yeah, millimeters was too big.
That's
I think
the
if
only
they define
size
it
that I'm getting, though. Like, should I know the sizes? Because it said on the study guide that I should know the relative sizes of them, and that's the only one that I'm getting is 0.2 to 20 micrometers for bacteria. Should I look up for the other ones, for the other 3 domains of life?
There's a nice figure that I have on page 1 of your Chapter 3 lecture slides, because this is microscopy now where they're talking about the size. And I just want you to know the relative sizes, not of how like bacteria compare with, human cell or a eukaryotic microbe like an amoeba or a virus. Right, so it gives you kind of the whole gamut of a virus, which is less than a micron, well less than it, and bacteria, which are about 1 micrometer or micron, and then some can get quite big, but those are exceptions. I just want you to remember it's about 1 micrometer. For bacteria, it's much smaller, on the order of so less than micrometers would be nanometers. So viruses are on the scale of nanometers. Usually the smallest virus, I think, is about 40 or 50 nanometers, but they can get up to 2 or 300, 2 to 400 nanometers. But they're like a good log order. So there's 1,000 times smaller than a micrometer is a nanometer. 1,000 times bigger than a micrometer is a millimeter. So millimeter, you're talking about these large amoeba, like a paramecium. It's even less. It's less than a millimeter. It's about 100 micrometer, but it's certainly much bigger than 1 micrometer, where you have for the typical bacterium. And then human cells fall somewhere in between there. Red blood cells a little bit smaller than a lot of our cells. A lot of our cells are more like 10 micrometers in size, but it really depends on what kind of cell you're talking about.
That's a good, I forgot about that slide. It's a good slide to go
back
to.
Another good slide is on page 2 of that set. I have even showing you, like the size of DNA, and then the virus, and then the E. coli, and the red blood cells, and a tick. So it's either of those figures give you an idea.
On this chat. It looks like we're the only one on this chat. Is it okay to ask you about the presentation that I have to do the proposal for?
Oh, sure. That's fine.
So we mentioned E. coli a couple of times already, and I don't know where I put my notes. Here they go. I was thinking about doing E. coli.